What does glucagon do?
Glucagon. Who wants to talk about it?
Isn't it the
Yeah, the
Opposite of insulin. Perfect. What is insulin?
what's that breaks down sugar?
Builds glycogen.
Okay, so how's insulin released? The pancreas? Where in the pancreas?
Beta cells.
Beta cells are two types of cells, right? There's Pacini cells. What do those do? This is the digestive section. Digestive enzymes, right? Amylase, protease, lipase, and those kind of things. And what activates those?
Cells in the
Just have a good review right here.
Like secrete.
Cells in the duodenum. Which ones? You got S, I, and K. I cells, right? What activates I cells?
Fat.
Any kind of fat, cholesterol, fats, all those kind of things, right? What is the hormone called that gets released from here? CCK, which then goes to the gallbladder, causes contraction, puts bile, liver secretion of bile, because these guys release the enzymes, right? And then stomach motility, decreased stomach motility. Good job, guys. Okay, what do K cells have to do with insulin? They release GIP, gastroinhibitory peptide. What activates them?
So,
Sugars are carbs, right? So we eat a bunch of carbs, and our body's gonna think what? It's gonna think I'm gonna have a big increase in sugar soon, right? So what should I release
Gastric inhibitory peptide.
to go and talk to what?
To the beta cells and make insulin.
Beta cells, right? Found where? What are these things called? The
Pancreatic isolates.
Yeah.
I don't know.
isolates of Langerhans. That like science that figured them out. I don't know if you didn't know that or not, but we talk about it in class. Like,
Yeah.
I just said it. So
Are
I'm
there
not.
Langer cells anywhere?
Yeah, that's what they're talking about. They found the isolates, they're alpha and beta cells. So beta cells are going to release insulin when they get activated by this guy, right? Or any kind of rise in blood glucose even. If you have a high blood glucose, it causes this to release too. There's just like a review from last section. So we release insulin in response to high blood glucose, and so then its goal is to eventually lower blood glucose. And how does it do that?
making glycogen.
Making glycogen.
Making glycogen or...
To make glycogen, what do we have to do though? This is like 264 status,
Yeah.
right? Glycolysis, remember this?
Yeah.
Where does glycolysis happen?
In cells.
In the cell. We have to get the carbs into the cell for this to happen, right? Through GLUT4. Remember how in the first part we had to make the decision if we're going to make ATP or if we're going to make glycogen? Remember that, the very first part of glycolysis?
Like,
Long time
Like
ago.
your second chapter
very,
of
very first part.
your second test, 264, right? Okay, so the whole point is to be able to have this cell, you have an insulin receptor, IR stands for insulin receptor. Insulin's in the blood. It comes along, looks like this, hits it, and then it stimulates GLUT4, right? It's on the surface, and what is GLUT4?
It's a co-transporter.
Yeah, it transports glucose into the cell, and then it can make ATP, or
Wait, I
glycogen,
thought
whatever it's gonna do.
GLUT2 transported in the cell, GLUT4 transported
Glucose
into
does
the...
transport.
GLUT2 does transport
Sorry.
glucose in the cell, but where?
Only in the liver and the brain.
And beta cells too, right? Because that's how we release insulin in the first place. You have a beta cell, glucose comes up to it, and there's a GLUT2 channel. GLUT2 channel allows glucose into the cell, right?
Which makes ATP.
Which makes ATP, which then
Stimulates potassium
the
sensitive. Potassium ATP sensitive voltage gateways.
to do what? Open or close?
Open.
No,
No.
doesn't it?
What do you
Wait,
think?
doesn't the
You're
glucose
close.
come in and block it?
Closes them,
It
because
closes
usually they're
them.
open leak channels, and so then the
Yes.
charge builds up.
Boom.
And will stimulate calcium channels to open.
Perfect. Good job. See, you know this stuff. Okay.
Okay, say that again.
So glucose comes into the cell and it can make ATP. You already know that, right? Then it can come over here and there's these channels that are potassium channels, okay? So these potassium channels are sensitive to ATP. and they will close if it gets it, okay? So if we close it, we get more separation of charge or less separation of charge in the cell? What do you think?
Less.
Because
More
potassium
separation.
leaves,
More separation of charge.
if potassium leaves, you get a positive out and a negative in, right?
Uh-huh.
And so that's a separation of charge. And so basically, if you close that gate, can potassium leave?
No.
So if it stays in there and then it doesn't separate anymore, and so you'll end up with less separation of charge.
Oh.
Does that make sense? It's really confusing, like this is, back to the actual potential chapter, right? 264, it's probably one of the more confusing chapters then. But it's, when you think about it, it kind of makes sense then. Potassium stays in here with his little negative friend, so it's less separation of charges. But that makes the inside of the cell more positive or negative? More positive, and it kind of gets towards this eventually actual potential part, right? The threshold will get closer to that, depolarization is what's happening. And then that will open up a calcium channel, it's a voltage gated channel. Calcium comes in, and what does calcium do to these guys?
It will bind the vesicles holding insulin and release them.
And it releases insulin to come out, right? Because they're these little guys.
So what does glucagon do?
So, the glucagon comes from alpha cells. Alpha cells release glucagon. Glucagon's whole purpose is to raise blood glucose. While insulin lowers it, glucagon raises it. And so if you don't have enough, so say you're fasting, right? Your body needs glucose, your brain needs glucose to function, right? You need energy. And so you've stored it in your liver, you stored it as glycogen, you have all these things. So glucagon is actually a hormone, it's not sugar itself. It leaves and it causes gluconeogenesis,
Which means the creation of glucose,
which means you're gonna make glucose from amino acids, or you can have
specifically in the liver, right?
Uh-huh. It's more especially in liver
So
for
it's
sure.
making glucose from amino acids, is that what you said?
Yeah, from proteins, right? And so if a diabetic has low blood glucose, then you're gonna want to give them a glucagon shot. If they have high blood glucose, you want to give them insulin, right? And so you have to kind of figure that out, because if you give them the wrong thing, you just cause the problem to get worse. Does that make sense? It's like if you give them insulin and they had low blood glucose, you would drop their glucose even lower, right? Put them into more of a coma, which we don't want to do. We want to bring them out of a coma, right? But normally you only give glucagon shots if they're like passed out and they're like not responding. Okay.
Okay, do T3 and T4 levels ever affect constipation or diarrhea?
I would say yes. It would decrease the motility. So you wouldn't,
Would decrease motility?
that's what I think. I'm not 100% positive on this one. I think it would, hypothyroidism could cause constipation possibly.
I think so.
Hypothyroidism.
Okay, that would.
Hyperthyroidism would cause you to run through food like there's no tomorrow.
Okay,
Because you metabolize like crazy.
okay.
Whereas hypothyroidism would slow down your metabolism. Whether this was on peristalsis or not, I'm pretty sure it would too. But that's just my thoughts.